All right, thanks. It's my pleasure to be talking to you guys today. I will be going over a synthetic biology workflow using SBOL Designer. So recently, ACS Synthetic Biology, a very well-reputed journal for synthetic biology, has announced their official support for SBOL as the data standard of choice for submitting genetic designs. So this means if you are a researcher and you want to submit a paper to ACS in Bio, then they recommend that you fill out the genetic design portion of your paper in SBOL. So I'll be going through basically a workflow that allows for these researchers to do this. So on the left, you see we have parts repositories. I'm sure you guys are familiar with some of them. So we have the JBase repository, which is actually hooked up to ACS in Bio. We have the iGEM registry, and we have a new one called the SBOL stack. And so we have parts in these registries, and then we need some way to connect them. So that is the job of sequence CAD tools. Examples of this are SBOL Designer and DNA Plotweb. And then we also have circuit computer-aided engineering tools like Trello and iBiosim that you can use to simulate and model these genetic circuits. So throughout all of this, we use a data standard called SBOL, the Synthetic Biology Open Language, to facilitate data exchange between these tools so that everyone can work on the same genetic design and you can have different parts of the workflow operated by different tools. So what is SBOL? There are two parts. First, there's SBOL Visual. So it's a visual standard so that when you read about these genetic designs in papers, you can have the same glyphs and be able to understand better. So for example, you can see like the bent arrow for a promoter. It's pretty much what biologists have adopted as the glyphs they want to use. And then we have the SBOL data model. So I'm sure some of you might be familiar with data models like FASTA and GenBank. So FASTA, you have basically just sequences, probably like passing around a Word file with a bunch of GATCs. And then you have languages like GenBank, where you have annotations. So you can say, OK, this bunch of sequences are a promoter. This is a terminator, stuff like that. And then SBOL 1.1 improves upon that by allowing a hierarchy. So you can define your genetic circuits with layered parts. So you can say you have this cassette, and then this cassette is comprised of a promoter, an RBS, a CDS, and a terminator. So SBOL 2.0 adds upon that, in that now you can have small molecules like ATC or whatever, and you can have proteins. So, and you can have interactions between all of these. So it greatly improves upon what you can do and what you can encode in these genetic circuits. Also, there is library support for SBOL 2.0. So I use libSBOLJ, which is also developed at the University of Utah. And there's also an online validator converter for SBOL 2.0. So if you design a file and you want to make sure that it's valid and compliant before you upload it to a registry, then you can do that. It's also hosted by Nona. So my work at the University of Utah so far has been on a tool called SBOL Designer. So it's a basic sequence computer-aided design tool that allows you to stitch together parts and create a sequence. It was initially developed at the University of Washington with Clark and Parja, and now I'm working on it. So a big goal for SBOL Designer was to update it from SBOL 1.1 to SBOL 2.0. So this largely, a lot of the data model has stayed the same, with like DNA components being the more generalized component definitions. But some parts of the data model have changed significantly. Examples of this are sequence annotations. Now, instead of just sequence annotations, you have sequence annotations, components, and sequence constraints. So I won't go into too much detail about what all of this entails right now, but I will be talking in the next couple of slides about how this connects to SBOL Designer as a tool and how you can use the tool to kind of like get a grasp at what you are manipulating. So that's the canvas. You can see on the bottom, you have a bunch of parts, these buttons down here, and you can click on them and drop them onto the canvas. So here we've dropped down like a promoter, RBS, a CDS, and a terminator. Each one of these parts, so like say the promoter, is actually comprised of a component definition and a sequence. So that's like the back-end SBOL data model that you are manipulating. But I don't want you guys to have to worry about that. All you can think about is it's a part. And that's abstracted. So when a biologist encounters this tool, they can just drop down parts and they know what that means. They don't have to worry about the underlying data model. So if you double click on a part, you can see that you can edit a lot of fields dealing with the part, and this is all, again, reflecting with the data model. So you have the role, and these are using a sequence ontology terms. So you can select from various roles, you can give it a name, a display ID, a version, a description, and you can attach a sequence. A big change with the backend data model, again, was sequence annotations. So now parts are ordered on the canvas using sequence constraints and sequence annotations. So sequence constraints define primitive ordering, so like part A precedes part B, and then sequence annotations define ranges. So you can say part A starts at this nucleotide and ends at this nucleotide. So I use both in the implementation for SBOL Designer, but again, you don't have to worry about it. They just are ordered that way. Another feature of SBOL Designer is the ability to do hierarchy with the new components that were introduced in SBOL 2.0. So you can think of components as references or links between parts. So we can say we have the left cassette there of the genetic toggle switch, and then if you focus in, there's a focus in button, you select it and you click focus in, you can see that there are actually 4 components associated with this component definition, or this part. And each of these components refers to those parts. So this is an organizational tool for designers, because then they can say, okay, we have this top level part, but it is actually comprised of these 4 smaller parts. And so doing this, you kind of have an easier way to organize and design your circuit. So here we can focus in again and say that box is comprised of a ribosome binding site and a coding sequence. So a new feature I've added that was not in the previous version of SBOL Designer is the ability to manipulate multiple designs in the same SBOL file. So say you have a bunch of designs and they're all related to the same project. Instead of having a single SBOL file for each one of those designs, you can actually put all of the designs into a singular SBOL file, and then you can basically treat it as a workspace. And then when you open that file, you can see that all of your designs are saved, and you can work on any single one of them and resave it back to the same file. Another feature I've added is the ability to import parts and import sequences. So if we go into the part editor and say we know what part we want because we've used it in another design that we have on disk or something like that, then you can click on the import part button, select the file you want, and then select the part you want from that file, and then it gets imported. So instead of copying, pasting, fetching the sequence, all of that, you can just import parts from existing files. So this really speeds up the design process. So another feature I've added is the ability to import from registries. So if you click on the little preferences button over there, you can navigate to the registries tab. Here you can select that you want to add new registries. So you can see here we have 3 here already, built-in parts, the SBOL stack, and a local repository. So built-in parts is a document containing all of the iGEM registries parts, and this comes pre-built with your installation of SBOL Designer. So you can select from that if you don't have a data connection or something like that. You also have access to the SBOL stack instances. So right now, this one is pointing to the Newcastle instance. So this SBOL stack, it's another repository, and it was actually developed at Newcastle University. And so I can fetch parts from that repository through the internet, or we can also point to local repositories. So if you have an SBOL file on disk and want to import parts from there, then you can also do that. So if you click Add, you can see that you can have a place where you can enter the name and the description of your repository, and then you can specify the URL or path. So if you want to point to an SBOL stack instance, then you would put in the URL for that SBOL stack instance. And if you want to have a local repository, then you would navigate to a file on disk and point to that using the path. So now that we have registries added, we can select a part and click on the little flashlight icon right there to import parts from a registry. So we're going to do it from the SBOL stack right here, because that's probably the most exciting one. So you select which registry you want, so the SBOL stack, and then you can filter parts by roles, and you can filter parts by name and display ID. And you can see it's actually in real time fetching all of the parts from the SBOL stack that match your criteria, and you can select one of those and import it. So here we have that promoter fetched from the SBOL stack over the internet, and it's now in your design. So that also really speeds up the design process. Another feature I've added is the ability to not only just save into a file, like overwriting, but also creating a new version. So this is really useful with the multiple designs feature, because then I can create a new version of a design that I've edited, and then save it to the same file, and then when I open that file again, I can see all the different versions of the same design, and I can see how it has changed over time, and such like that. And another feature I have added is the ability to export. So we support GenBank, we support FASTA, and we also have legacy support for SBOL 1.1. So SBOL Designer, you're designing the design in SBOL 2.0, but you could also export to any of these formats. So say we have this generic toggle switch designed in SBOL 2.0. We can export it as GenBank, what you see there, or we can export as FASTA. So I have a short little demo that I'm going to walk through that shows all of this being used to create something. So right now we're in a tool called iBiosim, which is also developed at the University of Utah. SBOL Designer is embedded inside of iBiosim as a plugin here, but it is also a standalone application. But for this, it's embedded. So we're opening an existing project, and this project contains a lot of pre-built parts for the generic toggle switch I'm sure all of you are familiar with. And so we're going to run through a little example of this workflow building the generic toggle switch. So I'll show you a part of the design process in each of these steps, but again, in the interest of time, I have a lot of this pre-cooked. So let's say we're going to build the TED-R inverter in SBOL Designer. You see you can click and drop in parts depending on what you want. So the TED-R inverter has a promoter, an RBS, a CDS, and a terminator. Here I am actually importing parts from the SBOL stack. So we can watch it again. Here I'll explain more about it. So this SBOL stack instance is hosted at Newcastle, and it's already added as a registry. You can see here. And then I can filter the parts, select which one I want if I know what the display ID already is. And I'm actually fetching this part from using the internet, and none of this is stored locally. So if you already know what parts you want, you can really just like drag and drop what you want into the design and then just fetch it from the internet. So it's very fast. You don't have to copy and paste sequences. You don't have to really search for your parts as much like you used to. Okay, so now I saved it, and now I want to use iBiosim. One of its features is to create an SBML model. So this is using another data standard called SBML, the Systems Biology Markup Language. And you can use this to simulate and model your designs. So here we create a species called TED-R. It's an input to this model. And then we have another one called black eye, which is an output. We set the initial concentration to 60. That's just it's a parameter for modeling purposes. And the red diamond you see is a promoter. So that basically represents the structural aspect of our genetic circuit, and we can associate with the SBOL that we just created in SBOL Designer. So we can select the TED-R inverter gene. You can see that's what we just made. You associate it, and then it turns into handy little SBOL visual glyphs, so you can see what you've associated. I've done the same thing with the lac eye inverter, but much of the steps are the same, so I'm not going to run through all of that again. But now that we have both the TED-R inverter and the lac eye inverter built, we can add them as modules in the top level genetic toggle switch. So here we are adding in the green rectangles. These are modules, so it's basically hierarchy. In iBiosim, we say we have the TED-R inverter and we have the lac eye inverter, and we are connecting it to all of its inputs and outputs, and we are also connecting it to states, to events, so that when we model it, when we simulate it, we can see the behavior and see whether or not it's correct. So that's what these little green boxes are here. They're events saying ATC and IPTG, which are the switches that control the behavior of the switch. We can say have them be high or have them be low at different time intervals. And then when we model it, we can see if the behavior of the genetic toggle switch is as we predicted. So right now, this is the analysis view, and we have it set to do a Monte Carlo simulation. We can click Run, and it will simulate. So after that's done, we can click on the graph and view the results of our simulation. So as you can see here, GFP, the green line, is turned on when IPTG is introduced, and then it's turned off when ATC is introduced. So it works. It functions correctly. So now to continue with the workflow, what if we want to upload this part to the JBay or ICE registry, and maybe we want to simulate, we want to attach it to a paper that we are submitting. So we can open part of this design, for example, the TEDR inverter gene, which we made in the previous video, and then we can export it. So you can see it also, we can export as FASTA GenBank and SBOL 1.1, but we'll export it as SBOL 2.0. And before we post it online and share it with the world, we want to make sure that this SBOL file is compliant. So if you go to sbolstandard.org, that's the website for our standard, and then go into the software tab, click on Converter Validator, you can actually select the file that you want to use, that you want to validate. You can do all of this through Nona as well. They have also hosted the SBOL validator. So you select the file, you say you want to output GenBank just to view it, and then you can click on Validate. And great, validation is successful, no errors. And then you can download the file and view it if you want. So you can see that's the actual GenBank that was just converted based on the SBOL file that we just uploaded. So it's pretty neat. You can do the same with FASTA again. So this validator converter was developed by a friend of mine, Zach Zundel, also at the University of Utah. Okay, so now we know we have a valid SBOL file. What if we want to submit it to a registry? So you can go to the JBay ICE registry, which is partnered with ACS SynBio. You can create a new part, fill in some fields, some details about it, and this is part of the genetic toggle switch. And you can upload the part. So I think the JBay ICE registry is actually using libsbol.j in the backend to manipulate these parts. So it's all one big community. And here you can see that the part is uploaded. It's been uploaded successfully. And there even is a handy little SBOL visual glyph right here. So you can make sure that the data, the contents of the file you just submitted are indeed the data that you want. So that ends the demo. So I would love any feedback, and I would love for all of you here to try it out. You can find the latest version of the SBOL Designer tool here at this website. You can feel free to post feedback or bugs, feature requests to my issue tracker on GitHub. That's here. And if you want to play around with iBiosim, there's also a release of that you can find here. So I'm anticipating the release of SBOL Designer 2.0 in about a month to coincide with the IWBDA conference happening at Newcastle. But yeah, I would love any feedback and for you guys to try it out before then so I can maybe make some improvements and iron out some final things. And with that, I'd like to thank all the people who have been involved with SBOL. A lot of these are Professor Chris Myers' students at the University of Utah, and they've all worked on iBiosim and SBOL in some point. And I would also like to thank the SBOL community in general for really paving the way for a data standard for synthetic biology. Without them, nothing would have been possible. And I'd like to thank all the organizations who support us. You could probably recognize some of the organizations here.